Hello everyone, my name is Rachel and I will be presenting for Group 1 along with Jean, Nicole, Thao, and Miguel. The topic of our presentation today is understanding the emergence of multidrug-resistant Acinetobacter baumanii and developing a multifaceted infection control plan. There has been an outbreak in our hospital. Patient 1 has been admitted to the ICU after a visit to the ER with severe pneumonia. The sputum culture has indicated the presence of Acinetobacter baumanii, which is resistant to all antibiotics on the screening panel. The following week, the microbiology lab isolates A. baumanii from patient 2 in the ICU with the same antibiotic-resistant profile. The next day, patient 3 shows sputum results with a positive culture for A. baumanii with a similar resistance profile. The ICU physician suspects that patient 1 has been the origin of the infection. Within the next 3 days, 4 more patients in the cardiac unit and the bone marrow transplant unit have tested positive for A. baumanii. The 5 of us have been tasked to develop a plan to isolate and stop the spread of the infection. In order to develop our infection control plan, we must understand antibiotic resistance and investigate the origins and transmissions of Acinetobacter baumanii. Jean will begin this section by discussing antibiotic resistance and gram-negative bacteria. Nicole and I will discuss the mechanisms of antibiotic resistance to 2 commonly used last resort drugs. Thao will cover the origin and transmission of A. baumanii. Miguel will introduce our multifaceted infection control plan. And finally, I will discuss how we intend to allocate funds for our plan, and I will also hold a brief Q&A.
First of all, what is antibiotics? Antibiotics are antimicrobial substances derived from microorganisms, and it is used to treat an infectious disease by inhibiting growth or killing pathogens. Penicillin was the first antibiotic successfully used to treat many serious infections, which was isolated from penicillin mold by Alexander Fleming in 1928. Thanks to Sir Fleming, this has become the foundation for treatment of infectious disease worldwide. And by using similar discovery and production techniques, researchers discovered many other antibiotics in the 1950s and 60s. The widespread use of antibiotics has extended average life expectancy and continues to serve as the main treatment for potentially fatal bacterial infections. However, there has been antibiotic resistance alongside the discovery of novel antibiotics because microorganisms find ways to survive and develop resistance to the new drug. So antibiotic resistance has become a challenging global crisis in recent years as it continues to increase in frequency and numbers. Antibiotic resistance is often referred to as a silent tsunami facing modern medicine. It can occur naturally over time, but the misuse of antibiotic accelerates the process of resistance because in many places, antibiotics are overused and misused in people and animals, and often given without professional prescription. Factors such as excessively prescribed antibiotics, availability of antibiotics without prescription, and inappropriate use of antibiotic drugs promote development of resistance. Environmental factors such as widespread use of antibiotics in agriculture and cleansing products can also accelerate antibiotic-resistant problems. In addition, there is increasing incidence of hospital-acquired infections, and they are becoming more virulent and more resistant to the antibiotics typically used to fight them. Another important cause of increasing antibiotic resistance is the selection of resistant bacterial strains by mutation and transfer of mobile resistance genes in bacteria. So why is antibiotic resistance a big deal? Why worry about it so much? The Centers for Disease Control and Prevention, CDC, estimates that each year in the United States, 2.8 million illnesses resulting in more than 35,000 deaths are caused by highly antibiotic-resistant bacteria, and many of these infections are caused by gram-negative bacteria. These resistant infections are harder to treat than those caused by non-resistant bacteria. And this challenges the ability to treat common infectious disease and make life-saving drugs less effective, resulting in prolonged illness, disability, and even death. The spread of antibiotic-resistant bacteria is a daily challenge for hospitals, with immunocompromised patients in the intensive care unit being especially at risk. According to International Nosocomial Infection Control Consortium and Sentry Antimicrobial Surveillance Program, multi-drug-resistant rates in gram-negative bacteria A. baumanii that cause ICU-acquired infections yield 63% and 57% respectively. Also, medical procedures and major surgery become very high risk with antibiotic-resistant bacteria. And all of these lead to increased costs of healthcare and place clinical and financial burdens on the healthcare system, patients, and their families. Infections caused by multi-drug resistant gram-negative bacteria have become a particularly serious challenge for healthcare professionals. Gram-negative bacteria differ from gram-positive bacteria with respect to the structure of the cell wall. They have a cell wall that has an outer membrane surrounding the peptidoglycan layer, and this outer membrane has lipopolysaccharides and lipids that provides antibiotic resistance to bacteria. As you can see in the diagram on the right, the main properties of gram-negative bacteria consist of three principal layers: the outer membrane peptidoglycan layer, and the periplasmic inner membrane. The largely impermeable cell wall of gram-negative bacteria is what makes them more resistant to antibiotics, which structure the gram-positive bacteria lack. As mentioned in previous slide, the outer membrane is an important barrier that excludes antibiotics from penetrating the cell wall, whose targets are beyond the surface layer. The outer membrane barrier can be regulated by environmental signals like antibiotics. Gram-negative bacteria can create resistance by altering the outer membrane such that mutations occur in hydrophobic properties or porin channels and alter the antibiotic efficacy. Other mechanisms of gram-negative antibiotic resistance include acquisition of enzymes that modify or destroy antibiotics, such as beta-lactamases, that mediate antibiotic hydrolysis and cause resistance to penicillin and cephalosporin. Efflux pumps is another key mechanism of resistant gram-negative bacteria that allows bacteria to pump out antimicrobial agent. Antimicrobial killing of gram-negative bacteria are also carried on mobile genetic elements transmitted to other bacteria by horizontal transfer of resistant genes, which I will be talking about in the next slide. The three mechanisms of horizontal gene transfer consist of transformation, transduction, and conjugation. Bacterial transformation is a result of direct absorption of DNA between two closely related bacteria where free DNA is picked up from the environment and naturally penetrate the bacterial cell membrane. In transduction mechanism, plasmid DNA enclosed in a bacteriophage is transferred from one bacterium to another. And this generates a lytic or lysogenic cycles, which involves reproduction and spread of the DNA. Conjugation is the main horizontal gene transfer mechanism for spread of resistance. It enables direct cell-to-cell contact where the conjugated plasmids make a connecting tube called pilus between the two bacteria. And through this pilus, genetic material can transfer from the donor cell to the recipient cell. So gram-negative bacteria can be intrinsically resistant, or they can develop various mechanisms to resist the effects of antibiotics and improve their chances of survival.
I'm going to present about Acinetobacter baumanii carbapenem resistance. Before I discuss the resistance mechanism, I'm going to briefly talk about beta-lactam antibiotics and discuss the mechanism of action. Beta-lactam antibiotics consist of penicillins, cephalosporins, monobactams, and carbapenems. So these antibiotics have a four-membrane, nitrogen-containing beta-lactam ring. But they differ in ring structure and attached chemical groups. In the picture on the right, you can see the different examples of these antibiotics and the base molecular structure of the beta-lactam. The beta-lactam ring is the key to the mode of action of these drugs because it is similar to the acyl D-alanine D-alanine, which is a substrate required for synthesis of the bacterial cell wall. So like I said, carbapenems are a class of beta-lactam antibiotics. They are capable of inhibiting beta-lactamases, which are produced by organisms like A. baumanii. They are considered a last-line agent because they have a broad spectrum of activity. It is used to treat severe or serious infections, and they are very stable against cleavage from many beta-lactamases. So what is the mechanism of action? The beta-lactam antibiotic binds to the PBPs or the penicillin binding proteins, which are enzymes involved in the formation of the peptidoglycan. It then inhibits cell wall synthesis so the bacteria can't form a peptidoglycan cell wall. The bacteria will have an ineffective cell wall which will lead to cell lysis. The mechanisms involved are divided into two categories: non-enzymatic and enzymatic. Non-enzymatic consists of efflux pumps, decrease in permeability of the outer membrane, and alterations in PBPs. The enzymatic mechanism involves synthesis of beta-lactamases. There are three groups under this category, but I will be focusing on carbapenemases because this is the most significant of the carbapenem resistance mechanisms. So how do beta-lactamases inactivate beta-lactams? The beta-lactamase is able to cleave the beta-lactam ring of the drug. This alters the structure of the antibiotic and inhibits it from binding to the PBPs. So what happens is that cell wall synthesis is able to proceed. So as I mentioned in the previous slide, carbapenemases is the most significant and versatile carbapenem mechanism. It is segregated into non-metallocarbapenemases and metallocarbapenemases. Non-metallocarbapenemases involves class A, C, and D beta-lactamases. It includes acylation and deacylation reactions. So what happens is that conserved serine in the active site of the enzymes attacks the beta-lactam amide bond. This results in an intermediate acyl enzyme complex. The intermediate is then hydrolyzed by the deacylating water molecule and forms the hydrolyzed product. The product is then released from the active site of the enzyme. On the other hand, metallocarbapenemases involves class B beta-lactamase, which is zinc dependent. It needs zinc for the reaction to happen or any other heavy metal. It involves water molecules that are coordinated to a divalent cation in order to activate and disrupt the beta-lactam ring of the drug. This mechanism does not form an intermediate like the non-metallocarbapenemases. These beta-lactamases, class A, C, D, and B, are responsible for the hydrolytic inactivation of different classes of beta-lactam antibiotics. So these enzymes are basically cleaving the beta-lactam ring to change the structure of the drug, so the drug becomes ineffective. So now when carbapenems don't work, the next and final choice of drug would be polymyxins, which Rachel will discuss in the next slide.
If a patient infected with A. baumanii is not improving with the use of carbapenems, the next and final resort is to treat with polymyxin antibiotics. These drugs are typically the last resort because they can be toxic to the patient's renal system and also have been known to cause seizures or neuromuscular weakness. The two most common polymyxins are polymyxin B and polymyxin E. Polymyxin E is also known as colistin. These potent antibiotics are generally given by injection. The target site of the drug is the lipid A portion of the lipopolysaccharide present on the outer membrane of A. baumanii. The two most common mechanisms of resistance against polymyxins that have evolved in A. baumanii both involve the target of the drug, which I had said on the previous slide was the lipid A portion of the lipopolysaccharide. Here on the right, you can see a photo of the structure of the lipopolysaccharide. The first mechanism of resistance against polymyxins involves the modification of the lipopolysaccharide. A single nucleotide polymorphism causes the addition of phosphoethanolamine to the lipid A. This modification decreases the negative charge of the outer membrane, thereby decreasing the drug's affinity to the bacterial membrane. The second mechanism involves the total loss of the LPS. The mutation is on a gene that is involved in the biosynthesis of the lipopolysaccharide. The complete loss of the LPS again causes a decrease in the negative charge and removes the target site altogether of the polymyxin antibiotics. These resistance mechanisms come with a potential biological cost to the microorganism. In vitro studies have shown decreased growth rates and virulence for strains of A. baumanii that show resistance to polymyxins. This cost is greater for the bacteria that have completely lost the LPS. Unfortunately, this same study found that there was no change in virulence or growth rates in vivo.
Acinetobacter baumanii is a gram-negative opportunistic nosocomial pathogen. It is a ubiquitous saprophyte capable of causing both community and healthcare-acquired infections. Although it is more commonly known for hospital-acquired infections, this pathogen can create a variety of problems, including septicemia, central line-associated bloodstream infections, ventilator-associated pneumonia. It can colonize and infect open wounds, causing wound sepsis, also endocarditis, meningitis, urinary tract infection. Its antimicrobial resistance and its propensity to cause such large, oftentimes multi-facility nosocomial outbreaks has led A. baumanii to being one of the top priority pathogens for new antibiotics development. Recent studies have shown that the two modes of transmissions are zoonotic transmission and occupational transmission, of which the occupational transmission is of more prevalence. Antibiotics have been widely used in livestock production to help with growth and fight infectious diseases. However, they have also been found to be associated with the emergence of the resistant A. baumanii. A few studies have been done to compare the A. baumanii animal isolates obtained from pigs and cattle to the A. baumanii clinical human isolates. And they found that the strains recovered from animal isolates lack resistance islands and class 1 integrons, which in contrast are present in many clinical human isolates. Both resistant islands and class 1 integrons play an important part in making the clinical human isolate, especially multi-drug resistance and aggressive in hospital settings. Class 1 integrons are genetic elements that are well known for its distribution of antibiotic-resistant genes in gram-negative bacteria. Resistant islands is a type of genomic island, consists of many antibiotic-resistant genes. Genomic island is a part of DNA segments that can be exchanged between cells to improve their survivability. However, the studies were inconclusive due to insufficient data and evidence collected to prove the relationships between animal and clinical isolates. Still, the possibilities of zoonotic transmission should be continually monitored, especially because of A. baumanii's capability to mutate and modify its gene rapidly. Occupational transmission, which is a more prevalent and confirmed mode of A. baumanii transmission, is also known as Acinetobacter because of its presence in soft tissues infections experienced by soldiers returning from Iraq and Afghanistan. Much of the transmissions of the bacteria to wounded soldiers seem to have occurred in US field hospitals in Iraq, where they're often faced with impaired hygienic conditions, which allow for A. baumanii to grow unchecked. The transmission of the bacteria onto wounded soldiers can also occur through healthcare workers in field hospitals who are also responsible for local Iraqi patients. The dry and sandy conditions of Iraqi deserts also act as an ideal environment for A. baumanii to thrive. This has become a serious problem for military and veteran hospitals, where soldiers returning back to the US from active duty in Iraq infected with A. baumanii are now treated in the same environment as other patients. This allows for A. baumanii to infect non-military patients as well. Therefore, it is critical to isolate and screen all incoming military personnel as well as military healthcare workers as an effective preventative control measures. One of the characteristics that make A. baumanii so resilient is its ability to form biofilms. Biofilms are aggregates of cells, complex mixtures of microbes enclosed in thick layer of polysaccharide. which make them resistant to antibiotics and therefore extremely difficult to eradicate. Biofilm is mediated by an adhesive organelle called CSU pili. This helps A. baumanii to stick tightly onto different surfaces so that the formation of biofilm can occur. CSU pili have proteins that form these finger-like projections that help A. baumanii to latch onto these hydrophobic cavities in both biotic and abiotic surfaces. The protective layer of biofilm allow A. baumanii to sustain and resist through many different surface cleaners and different environmental stress, such as disinfectants, detergents, phagocytosis, desiccation, as well as antibiotics. The outer layer of biofilm form a physical barrier, making it difficult for antibiotics to penetrate. And so that allows the cells within the biofilm to survive the antibiotic exposure and therefore can cause recurrent infections. Also, because there's like a lack of nutrients within the biofilms, so that can slow down the metabolism of the bacteria. And many antibiotics require bacteria to be rapidly dividing for it to be effective. So the slower metabolism can actually reduces the effect of the antibiotics. A. baumanii's biofilm forming ability is especially effective in hospital settings where there are favorable surfaces like patients and medical equipment such as mechanical ventilators, catheters, respiratory care equipment, dialysis machines, hospital furniture, and healthcare providers' PPE. The bacterium's increasing capacity to develop resistance against the strongest antibiotics while surviving for weeks on hard surfaces, as mentioned, can lead to an increased colonizations of the bacterium in hospital environments. Increased hospital-acquired infections can result in increased morbidity and mortality. Those in intensive care units seem to be most affected by the bacterium because they're usually the ones with weakened immune system or hooked to ventilators for long periods of time, deep tissue damage, and on respiratory therapy equipment and indwelling catheters. A. baumanii transmissivity is also attributed to a process called horizontal gene transfer, in which it can acquire and edit favorable genes from other bacteria. So not only does it possess a number of resistant genes itself, it can also accept resistant genes from other bacteria. A study shows A. baumanii uses all resistant mechanisms such as conjugation, transformation, and transduction to successfully resist and survive against carbapenems, which are one of the classes of antibiotics used in A. baumanii's infections. Its ability to form biofilms on both biotic and antibiotic surfaces, along with its ability to adapt and change through horizontal gene transfer, has given it an outstanding survival properties. It allows it to grow persistently in unfavorable conditions and environments, and also help with its effective transmission. A. baumanii hospital outbreaks have also been linked to healthcare professionals with poor personal hygiene who neglect proper hand washing and sanitation protocol. They can become carriers of this opportunist epidemic strain. Family members who interact with these professionals can also become potential carriers. Besides hospital staff, visiting friends and relatives or patients who also introduce external strains of A. baumanii. This again exposes the pathogens to optimal conditions for effective colonizations. A. baumanii's ability to form biofilms and continuously changes its genetic makeup through HTT allows it to combat antibiotics continually and successfully, making hospital infection exceedingly feasible.
Here we summarize how to deal with the multi-drug-resistant Acinetobacter baumanii, pictured here on the right in purple. Because patients who are immune suppressed, victims of major trauma, incubated with catheters or mechanical ventilators, they are susceptible to infections with A. baumanii, requiring the development of a plan to eradicate it from high-risk units. First, the adoption of an active surveillance program to monitor current and new infections that are potentially entering the critical units. Those patients that are positive for A. baumanii will be cohorted into an isolation section in each respective critical unit, Environmental decontamination will become routine in all critical units in order to reduce cross-contamination with healthcare workers and other non-infected patients in each unit. Additionally, patients in isolation that are either colonized or infected with multi-drug-resistant A. baumanii will undergo decolonization procedures. Finally, the different facets of the infection control plan will come together with staff education practices to promote compliance. The program screens patients entering the various high-risk units for all multi-drug-resistant A. baumanii. Additionally, any patient presenting a new case should have everyone else that's in proximity to the infected person tested to ensure it didn't spread. Outbreaks around the world have used rectal/stool and throat/pharyngeal swabs to detect colonization or infection. Other sites that are screened include wound and catheter sites because they increase the sensitivity of detection, and those patients that are most susceptible to infection via their exposure to the environment, such as patients on ventilators. Recall that A. baumanii can form biofilms on glass and plastic surfaces and resist desiccation, making it a formidable enemy in infection control. To identify sources of the outbreak in the environment that may lead to nosocomial infections, we take swabs of different potential fomites, such as doorknobs, bed rails, bedside tables, and vital sign monitor touchpads, sinks, infusion pumps, and floors in patients' rooms. The screening validates whether or not cleaning done by healthcare workers and other methods have successfully removed traces of A. baumanii. Next, we test the swab samples from the active surveillance program to be enriched in media and then subcultured onto McConkey plates for A. baumanii isolation. Notice the non-fermenting characteristic of A. baumanii on the right. In our infection control plan, we'll use the MALDI-TOF for identification of A. baumanii. Considering that we will be processing a lot of cultures during our outbreak, we found it useful to use MALDI-TOF for its high throughput, speed, and reliability for identification. After identification of A. baumanii, we perform susceptibility tests that demonstrate criteria that allow A. baumanii to be considered a multi-drug-resistant organism. The susceptibility tests will produce MIC values for various antibiotics shown here. The breakpoints which decide whether a bacterium is susceptible or resistant is defined according to the CLSI guidelines. Depending on the breakpoint values, the bacteria will be classified as susceptible, meaning it will not grow in the presence of the drug, or resistant, meaning it will grow in the presence of the drug. Intermediate classification falls in between those two. CDC rules that in order for A. baumanii to be classified as a multi-drug-resistant organism, it has to be either intermediate or resistant to at least 3 antibiotic drug classes shown here, such as carbapenems, aminoglycosidases, and cephalosporins. Because multi-drug-resistant A. baumanii is a nosocomial outbreak, it poses a risk to vulnerable patients in high-risk units. Infected/colonized patients are separated from the remainder of the patients in an isolated section of their respective unit. Healthcare workers from all different backgrounds, including doctors, nurses, environmental services, respiratory, and physical therapists, must adhere to contact isolation precautions, meaning hand hygiene before and after interacting with isolation patients. Additionally, wearing gloves, gowns that are disposed of after interaction with these patients. Applications of disinfectants to commonly used items, such as clinical devices including thermometers and stethoscopes, and in-room devices such as infusion pumps and respirators, will be disinfected using quaternary ammonium compounds found in sandy wipes or other disinfectant wipes. Other effective products used at other hospitals include bleach products. Other common items such areas that must be disinfected include bed rails bedside tables, and mopping the floors with previously mentioned germicidal disinfectants. As a terminal disinfectant, we're investing into a portable hydrogen peroxide treatment of rooms potentially contaminated with A. baumanii. This technique is a relatively cost-efficient method to provide sterilization of entire rooms within high-risk units. This method is promising because studies have shown results of negative cultures both in 24 hours and in 1 week after decontamination, leaving behind no residual biocide. Additionally, the National Health Service in the UK considers it the gold standard to room decontamination. Another measure to prevent further dissemination of A. baumanii in various units is to use daily whole body antiseptic washes on colonized/infected patients. Studies have shown that patients tend to contaminate their environment and remain persistently colonized throughout their visits. The idea here is to help colonized/infected patients reduce the number of A. baumanii bacteria on their skin in order to reduce the opportunity for cross-contamination of others in the hospital. Here we present two antiseptic body washes, chlorhexidine and octinidyne. Using 4% chlorhexidine on colonized/infected patients has shown significantly to reduce A. baumanii colonization with whole body washes. However, some patients have reported allergies to its use. Another option that is already used for MRSA patients is octinidyne, which has similar effects of decolonizing patients. In order for all the previously mentioned infection control plans to work more efficiently, we recommend weekly meetings among leaders, including doctors, nurses, environmental services, and other healthcare workers to discuss the conditions of each high-risk unit, education, and compliance monitoring. Through active communication, we can mitigate issues such as overlooking responsibility of duties among nursing staff and environmental services. Training healthcare workers in proper contact precaution techniques can be aided with education programs and videos that should be mandated for working. Education should emphasize how healthcare workers can potentially cross-contaminate themselves and other patients by not adhering to proper hand hygiene and proper PPE use. Hiring infection control specialists to train healthcare workers from different units can help us promote compliance. Infection specialists can monitor healthcare workers for proper time guidelines for disinfection, hand hygiene, and proper PPE use. Studies have shown that behavior does improve when healthcare workers are being monitored. We suggest hiring healthcare workers from different high-risk units to monitor other departments in order to reduce bias. This concludes our infection control plan for the outbreak of multi-drug-resistant A. baumanii.
The 5 of us were given $1 million to implement some of the infection control measures that Miguel discussed in the previous section. The first thing we wanted to invest in for our 1-year plan to contain multi-drug-resistant A. baumanii was the Bruker MALDI, which is an instrument that is used for rapid and efficient bacterial identification. The cost for this instrument is $250,000, with another $70,000 for maintenance, reagents, and supplies for the entire year. We also need disinfectants to decontaminate the rooms of infected patients and commonly touched surfaces. Bleach can cost up to $170 per day, and wipes to go in every single room can cost up to $500 today, which brings us to a total cost of $245,000 for disinfectants for the year. We will need extra PPE for hospital personnel and for patients that are carrying the pathogen. This will cost another $250 a day, bringing us to $91,250 per year for PPE. We also plan to hire 3 infection control specialists, 1 for each of the infected wards, including the ICU, bone marrow transplant unit, and cardiac unit. These specialists will train and advise hospital staff during the outbreak. Each specialist will be paid a salary of $70,000 per year, which is standard. Finally, we will invest in a VHP 1000 ED Steris which is a machine that uses hydrogen peroxide to sterilize rooms that have contained infected patients. This machine and reagents will cost $100,000 for the year. This leaves us with $33,750 remaining, which we will use in case of an emergency. So here we have some questions that you should know by the end of this presentation. Number 1, what is the main mechanism for spread of resistance genes in horizontal gene transfer? The answer to this question is the conjugation method. Number 2, what are the 2 last resort antibiotics for treating Acinetobacter baumanii infections? The answer to this is carbapenems and polymyxins. Number 3, how do beta-lactamases deactivate beta-lactam antibiotics? The answer is they cleave the beta-lactam ring of the drug and alters the structure of the beta-lactam. Number 4, what are 3 parts of the infection control plan? The 5 possible choices are active surveillance programs, cohorting patients, environmental decontamination, infected or colonized patient decolonization, and staff education and compliance. Number 5, what is one of Acinetobacter baumanii virulence factors that contribute to its resiliency? The answer is biofilm. So here's our list of references, and I just want to note that these references are only for the breakdown of costs, and our other reference list was just too long to include in this, but that is in our paper. And I want to thank you all so much for listening. I hope you have enjoyed our presentation about multidrug-resistant Acinetobacter baumanii.